Thank you, Richard, for that brief introduction. And hi, everyone. This presentation actually is intended to give an overview about regarding a high-content, high-throughput technology that was discovered here at the CSIR by Dr. Neil Emmons. But before I proceed, I would like to give a brief history about the inception of this technology. I'm going to talk about the different platforms that have been used in screening. I'm going to give a comparison between these different platforms. And I'm going to talk about the RNAi platform, as Dr. Richard mentioned, that was discovered here at the CSR, and how we validated this technology, and how we intend to use this technology for potential applications. And I'm going to summarize to give the long-term goals behind this technology. Over the past 2 decades, there has been extraordinary advances in the field of functional genomics. And 2 of these events that contributed significantly to these advances are the discovery of the ribonucleic acid interference mechanism by Nobel Prize winner Fire and Mello, and secondly, the sequencing of the human genome. I'm sure most of us here, if I take into consideration that we are scientists, we are familiar with the central molecular dogma. But for the non-scientists, this is simply the flow of genetic information from the gene bank to the production of the protein, then to the phenotype. Most of our cellular system is controlled by this molecular dogma. However, there are times where the system tends to produce an unwanted protein, and most systems now have devised a means to inhibit the production of specific proteins, then term-to-term RNAi mechanism. What the system simply does is it synthesizes small interference molecule known as the siRNA that goes to bind to the RNA and destroys the RNA, thus inhibiting the production of the protein. The second point actually I made was sequencing of the human genome. There are hundreds of thousands of chemical molecules encoding for the human genome that is currently being synthesized, such as the siRNA, the mRNA, the shRNA, and the CRISPRs. And scientists now are currently harnessing this, the RNAi mechanism, coupled with the sequencing of this human genome now to interrogate biological processes and also to understand the role played by genes in host pathogen infection. The question that remains is, how do we screen through hundreds of thousands of chemical libraries with relative ease and in a short period of time? There have been a lot of platforms to screen through these libraries. One of them is the traditional method, whereby they use 96 word plate, 384 word plate, 1536 word plate. However, manual handling of this plate is very laborious and time-consuming. So most institutions now are using highly automated liquid devices that are fully integrated system and requires huge facilities and the experience of very trained workers, making this platform relatively very expensive. So there is a rising need now for a high-throughput technology. So currently now, the most researchers are adopting the microarray technology. Like the conventional method I showed to the left here, these are specialized pins that actually is being used to print spots, chemically spots on solid supports. Despite the high throughput nature of this platform, they are relatively very slow, printing approximately 100 arrays in 17 hours. Because after each cycle of print, the pins are being washed, blotted, air dry. And also the pins actually in this conventional method has relatively very small reservoir volumes, taking approximately 100 nanoliter. Also, there is quite a lot of waste in this process because of the nature of the encapsulation mixture that is used for the printing. Because the pins are blocked during printing and also prevents the printing of accurate spots. The pins used in this technology is very expensive, and because it's an assembly that is used for this conventional method, this platform cannot be scaled up. So to overcome such limitation now, CSIR has developed a standalone capillary-based printing technology. Like the instrument you see to the left here, this was actually built here at the CSIR. This is a capillary-based technology. And with this platform now, we can measure printing quality using a single continuous print of siRNA samples. And our print quality is greater than 95%, which is above any benchmark in any high-throughput screening platform. And we are capable of printing 500 to 750 essentially identical arrays in 2.5 hours and using just 2 personnel, making this platform relatively very expensive and also easy to operate. Just to give you a comparison between our approach and the conventional approach, which is kind of similar to our approach. Our approach, we use capillary-based printing, while in the conventional approach now, they use specialized pins. The microarray pins used in this current technology is not a solution for high-content screening because they have a relatively very low reservoir volume. They are very expensive. See, one of those pins costs around $750 each, as compared to one of our capillaries approximately less than 10 Rand. And then they are not suitable for the transaction mixture because this mixture actually is kind of viscous. They are very slow, and as I earlier said, they are wasteful in the mix. Meanwhile, with our capillary-based printing, capillaries deposit accurate spots. Our reservoir volume is 40 times compared to the conventional method. The capillaries do not need refilling or washing like in the case of the conventional method. They fill automatically by capillarity action. And there exist different sizes of this capillary. They're inexpensive, and also they're commercially available. You can get it basically anywhere. So just to give you an overview of our platform, how we print. So you have your encapsulation sRNA solution, and hopefully I have it here. You have your capillaries are supported on a metal plate like this one. It has 36 channels that the capillaries sit into it, and this plate is now mounted onto a loading duct. And using a microfluidic plate like this, the ones here, that I show you, you fill in your encapsulation solution, and the capillary now filled by capillarity action. And each of these plates now is now stuck into a head to form an assembly of array. And we can print basically 3,150 spots in 2.5 seconds contact time. Just to reiterate my point here, to compare the CSL array and the conventional array, you can see that we are capable of producing 3,150 spots in 2.5 seconds compared to the 24 spot that is being printed by the conventional method because they have an assembly of pins. We can produce greater than 1,000 arrays in 1.5 hours as compared to 100 arrays that is produced by the conventional method in 15 to 17 hours. And our platform is relatively very cheap as compared to the conventional method, and also it is easier to use our platform. Anybody can use it. Just 2 personnel can operate this platform, but with the conventional method, it's a highly automated system and requires the expertise of trained workers or very experienced people. And also to compare our platform with, let's say, 96 well plate, our platform can print encapsulation spot on this high-density plate, and we're capable of printing, as I earlier said, 3,150 spots. And one of our plate is equivalent to 32 96 well plate. It's easier to use one plate rather than using 32 96 well plates. And we have one of our plates is you can compare it to 3 84 well plate and 2.5, let's say 2.05, 15 36 well plate. However, in most screening facilities, they don't use the 384 well plate and 1536 well plate because of the volume that actually is being used during screening. And also, treating our high-density plate is like treating one well in the 96 well plate. And since we can print 3,150 individual sRNA on our plate, we do expose this sRNA to the same environmental condition, and as opposed to treating the 96 well plate separately, thus reducing the variation during your bioassays. And also, it is easier to handle one plate rather than handling 32 96 well plates. Okay, our research. We do print optical addressable spots that measures between 350 microns in diameters. And each of these spots now has a specific sRNA that silences a specific protein. So we cultivate cells on this sRNA spot that is being printed, and the cells now takes up the spot together with the chemical that is in the spot and silence the specific genes of interest. The two slides that I'm going to show you now is just going to show you gene silencing. So here is a slide of gene silencing. We used two sRNA, a non-coding sRNA and also a P65 sRNA. P65 is simply a transcription factor, actually that occurs in basically all cellular processes. This image this is a non-targeting sRNA, and this is the P65 sRNA spot. The green background that you see there, that is a P65 expression. So if you go now to the next image here, you can see the green background of P65 expression around the non-targeting sRNA, but when you come to the P65 sRNA, you can see that there is no expression around this area, telling us that the P65 expression has been reduced by the P65 sRNA. Then the next example is the INCHEM, which is actually a regulatory factor of the chromosome passenger complex. And this INCHEM actually helps to complete cell division during mitosis. And if you tend to silence this gene now, cells are going to form multiple nuclei because there's not going to be complete cell division. So we printed an INCHEM spot and also a control spot. To the left, you have your control spot and you have your INCHEM sRNA spot. And as you can see, you see cells with double nuclei in this spot with INCHEM, showing that the INCHEM sRNA has silenced the INCHEM gene. After this proof of concept that we could actually use our technology, we could actually print sRNA encapsulated spots, and we can actually show that this spot will silence a specific gene of interest. We went further now to validate our technology. So we validated the technology using a well-characterized signaling pathway, which I guess Dr. Schofield also did mention, the NF-kappa-B signaling pathway. That is a well-characterized pathway. Okay, here, the assay conditions here were designed to give maximum P65 transport into the nucleus. But however, our goal here was actually to determine the genes that actually are involved in this pathway. So, P65 actually was detected in the HeLa cells using indirect immunolabeling after TNF stimulation. Here you have your unstimulated cells, whereby the green background is a P65 expression. You see, the P65 now is located in the cytoplasm of the cell. But once the cell now is being stimulated with the TNF-alpha, most of the P65 now runs into the nucleus of the cell. Now, for our experiment, we printed a sub-library of the human genome. And this library comprises of all the kinones. And during our print, we incorporated some controls like the non-targeting, the exporting, the P65. And these standards were actually introduced now to measure the print quality during our experiments. And we printed a total of, sorry, why do I keep doing that? And we printed a total of 3,150 individual sRNAs. Okay, for our gene extrapolation now, to locate for our gene of interest, we use two known genes, actually, that influence P65 nuclear transport into the nucleus, which are actually CHOC and IKB-KB. Both are kinases regulating signaling from TNF now to NF-kappa-B. And loss of these kinases stop nuclear import of P65. And both kinases were actually incorporated into our print. We did 32 replicates of this screen, the highest ever known in most screening. I've gone through so many screening papers, and basically most screeners use between, let's say, 3 and 6 replicates. But we did 32 replicates of this screen. The data actually of these screens were now normalized, and the median values now were projected into a PCA. And PCA is simply a technique that actually emphasizes variation and bring strong pattern in data sets. Okay, let me speak English. Okay, let's assume actually that we designed an experiment actually to separate the different racial group. And let's consider ourselves in this whole like a data set. Once we are being projected into the PCA. And what the PCA does is it will take the black and put into one position, the white into another position, maybe the Indians and the Chinese in another position. So that is how it works here. So our data now from our 32 replicates after normalization and the median value was projected into the PCA is separated the data based on the specific phenotypic expression. You can see here our control value, all our control, for example, the P65 actually is in one location. You have your exporting and the toxic compounds in another location. And then empirically now, we use chalk and IKB-KB now to select for our candidate genes. We selected 51 candidate genes, and we blasted these genes against existing data, means data that has already been produced, and we had greater than 50% connectivity. And definitely, the rest of the other genes now are potential candidates that regulate the NF-kappa B signaling pathway. The image of you on this side is just an image showing the expression after this, the cells were exposed to different spots. So to conclude now, we've screened the TNF-alpha pathway at the highest density ever performed to our knowledge. We've performed greater than 100 individual cell-based experiments in 4 days and 2 extra days to analyze. And we actually performed using high replicate. We did 32 replicates, making our system very robust. And using conventional method, like the one I mentioned earlier, is going to take you 3 months to do the same experiment. And therefore, our platform, the CSR Sonics array technology, is suitable for high-throughput screening. This technology now is currently being commercialized under the name Sonics in Boston, and their first product line was launched last month, and this was an event that was attended by the Director of the Biosciences and also the Communication Department. And as you can see here, this is Dr. Sibisi and Richard Ferron being mentored on how to print arrays. And here you see Dr. Neil Emmons, Dr. Sibisi, and Richard Ferron. And Dr. Neil Emmons is the main architect behind this technology. He's the one running the facility in Boston. And here I am trying to rub shoulders. Okay, where do we move from this point onwards? We've shown that actually sRNA is the most challenging format of high content screening. But having proven that we, in the CSIR and in Boston, we can easily print this sRNA, we needed another venture. So now, we want now to adopt this technology now to produce compound arrays, which is actually a new architecture. Compound arrays have very great potentials. However, this platform has already been shown by Bailey and Co. that it's possible to print compound arrays. Compound arrays have not been adopted because of the technical complexity of the nature of the compounds. They have different compounds, let's say hydrophobic and all those type of things, hydrophilic, different molecular structure and all those type of things. And Sonics array was developed for mass production of compound array. I'll just go into the next slide, and I'm just going to give you a brief proof of concept that was performed by Bailey to show that you can actually print a compound, and this compound can actually have an effect. So in this paper now, Bailey printed PAO, which is a toxic drug, that's phenylaxine oxide, onto a chemically modified glass slide. And he actually cultivated cells on this slide, and as you can see the result, where you had your PAO spot, you can see that there's no cell there. The PAO was released by the polymer because he used the polymer to encapsulate this drug, and the drug was released in a timeless manner, and it killed the cells in the periphery of the spot. So our first objective now was trying to replicate this Bailey's method using our platform, which we did, of course, and we had better result anyway, because in Bailey's paper, actually the spot were not optically addressable. We could actually make our spot optically addressable. And in Bailey's method, as I earlier said, he printed layers that he had the drugs, the polymer, were all printed in layers. But in our case now, we actually were impregnating the polymer with the drug, and now we print this mixture onto a glass support. And as you can see here, you have your cells that actually is cultivated on the spot, and everywhere you see your your PAO spot, you see that actually the drugs is being released now and kills the cells around the spot. The other spots are just control spots. There's no drugs in those spots. And this therefore shows that our platform is capable now. We can use our platform now to print compound arrays. Okay, to conclude, us at the CSIR, we just want to create a miniaturized platform whereby scientists can use to rapidly print through small or natural compounds. This platform is to offer South Africans and African researchers a high-throughput screening product that is miniaturized and standardized, and we want to create a commodity that is portable, that is easy to manufacture, that is out of the box, that exceeds current technology, and that's also to create an outstanding product. This will enable researchers to access rapidly to test thousands of molecules in various bioassays in an easy and time-use manner. That is screening through hundreds of small interfering molecules, small molecular compounds, and also to look for alternative uses of already FDA-approved drugs, and finally to screen for cDNA. So what is our overall goal? Our overall goal now is to create an easy-to-use commodity that researchers can use to screen for biological targets that will serve as lead chemicals for new therapeutic drugs. Thank you.